Assalamualaikum. My name is Fatin Aina binti Hamzah. I'm from AS257. I'm supervised under Dr. Aziah binti Abdul Aziz. I'm going to present about my research SCCmec typing in clinical isolates of Staphylococcus epidermidis. This is my table of contents: introduction, literature review, third research methodology, result and finding, lastly conclusion and recommendations. Look about introduction. Research background. The connection of the methicillin-resistant gene MAC-A gene with antibiotic is strong. Second, in S. epidermidis has been found to harbor SCCmec type 1, 2, 3, 4, and 5. Problem statement. The presence of MAC-A gene in S. epidermidis has caused resistance towards antibiotics. Identification. Local isolates of S. epidermidis carry MAC-A gene and what type of SCCmec typing they are harbored. Significance of study. S. epidermidis isolates may contribute to the genome flexibility, provide an advantage in bacterium's quick adaptation to changing environmental conditions and genetic pathway. SCCmec related to the transfer of antibiotic-resistant genes to another Staphylococcus species. The objectives of this experiment: the first one, to identify MAC-A gene in the S. epidermidis clinical isolates. Second, to determine SCCmec typing in S. epidermidis clinical isolates. Moving on to literature review. S. epidermidis is coagulase negative staphylococci which enable to produce coagulase enzyme. It is also a commensal human microbiota but can turn to an opportunistic pathogen. S. epidermidis can cause clinical infection, neonatal sepsis, prosthetic joints infections, and peripheral or central intravenous catheters. Let's take a look about antibiotic-resistant properties. The first one, MAC-A gene in SCCmec. According to Pinho on 2001, responsible S. epidermidis resistant to beta-lactam antibiotic such as methicillin. Next, the MAC-A gene is responsible for encoding penicillin binding protein 2A. The figure shows here is the structure of MAC-A gene. Next, staphylococcal chromosome cassette SCCmec is a mobile genetic element that contains methicillin resistance gene, MAC-A, and other functional genes. There are two essential components in the SCCmec, the MAC gene complex and the cassette chromosome recombinase CCR gene complex. This figure shows the structure of SCCmec, which is half MAC gene complex and CCR gene complex. SCCmec is transmitted among the S. epidermidis and S. aureus. According to Hansen and Solid 2007, type 3 SCCmec has been reported in clinical and community isolation settings. SCCmec type 4 is often isolated from S. epidermidis, both in the community and in the hospital setting. In the previous report of Wisplinghoff on 2003, horizontal gene transfer of MAC-A gene in S. epidermidis between staphylococcal species. Community acquired methicillin resistant S. aureus CA MRSA gain additional mobile genetic element from S. epidermidis via horizontal gene transfer. Hence, this gives S. aureus to survive during infection or resist antibiotic treatment. Moving on to research methodology. Verification of clinical isolates. isolation of S. epidermidis. I was isolates on mannitol salt agar MSA plate to differentiate from S. aureus. The positive and negative control used were S. aureus and S. epidermidis. Biochemical test, catalyst test to differentiate S. epidermidis from Streptococcus sp. Hence, I used the control for negative and positive Streptococcus pyogen 80 and S. epidermidis 8. Then, coagulase test to characterize S. epidermidis as coagulase negative bacteria, S. aureus as negative control, and S. epidermidis for positive control. And last but not least, gram staining. The control used to indicate negative result E. coli and positive result was S. epidermidis. To detect make agent according to objective 1, first one, DNA extraction performed using Cajun kit according to the manufacturer's instruction. After that, DNA amplification, which using single plex PCR. This is the primers that I have used, and the PCR protocol that I have used based on Boyer 2007. After that, gel apheresis, which is 1 microliter DNA ladder and DNA extract of 5 microliter and 1 microliter of loading dye. And then the mixture was transferred into the wells of the gel electrophoresis and run at 85 voltage for 16 minutes. For objective 2, to determine SCCmec type 1, 2, 3, 4, and 5, DNA extraction performed using Cajun kit according to manufacturer's instruction. Then DNA amplification, which was used multiplex PCR, and this is the protocol cycling that I have used also from Boyer 2007. And then gel electrophoresis and run for 16 minutes. Results and finding. Verification of clinical sample growth on MSA plate. As we can see from figure A, S. aureus, which the yellow color. For figure B, S. epidermidis, red color. And the 6 clinical sample tested displayed red color on MSA, which verified as epidermidis. Coagulase test. No clump was observed on the slide of S. epidermidis or the clinical samples. Hence, all the 6 clinical samples were S. epidermidis. Gram staining. Figure B shows S. epidermidis positive control under gram staining that showed cocci. Also for C, B13 representative sample under light microscope. And all the 6 clinical samples show the positive result, which were S. epidermidis. Moving on to detection of MAC agent. This figure here show 500 pp for ladder and 533 pp for expected bands MAC agent positive. S. epidermidis clinical isolates, only 5 out of 6 samples were confirmed to carry the MAC agent. However, B18 clinical sample consider methicillin susceptibility Staphylococcus epidermidis (MSSE). Methicillin resistant for S. epidermidis found was 75 until 90% from nosocomial infection, which greater than S. aureus isolates. For the resistance, Next, moving on to identification of SCCmec typing. The figure shows here SCCmec type 1, 2, 3 was positive control for SCCmec type 4, positive control B13, 15, 16 and 17. However, non-typable was B14. SCCmec type 3 and 4 were predominant in clinical samples. SCCmec type 4 of S. epidermidis might be a reservoir to community acquired methicillin resistant S. aureus (CA-RSA). S. epidermidis, SCCmec type 4 clarified that 98 until 99% similar to SCCmec element identified in SCCmec type 4 CA-MRSA strains. In contrast, Ito in 2001 proved that 36% isolate S. epidermidis SCCmec type 4 found around the 1970s. Meanwhile, between 1981 and 1985, only 2 out of 38 MRSA isolates from patients carrying SCCmec type 4. Last but not least, conclusion and recommendation. The objective of this experiment was successfully achieved to identify the MAC agent and determine SCCmec typing in clinical isolates of S. epidermidis. The MAC-A and SCCmec type discovery can improve infection control and produce new antibiotic treatments for future. Recommendations. The research may be conducted on a large number of clinical samples of S. epidermidis. Furthermore, this research may extend to SCCmec type 11 in S. epidermidis. All of these are references that I have used in this research. Thank you. All of these are references that I have used in this research. Thank you.